So my name's Joe McKenna, and I'm a researcher at the University of Warwick, and I'm Deputy Chair of the Cells Panel of the SEB. And in this talk, we are in for a real treat. We're in for a very, very good talk here. So we've got Dr. Oliver Castell, who's from the University of Cardiff. Oliver is a serious brain power early career research fellow, which is very nice title. And he's a senior lecturer in the School of Pharmacy and Pharmaceutical Sciences. Oliver was the Cell Section Medal winner in 2016, and he's come back to give us an update on how things are going. And Oliver's going to give us a really, really useful talk around his career and his research, and from early beginnings all the way through to where he is now. So he's a pharmacist by training. He did his PhD in Cardiff, and he explored the unique characteristics of flow on the microscale, high efficiency chemical separations. And he was awarded the Destiny Memorial Award for Innovation in Separation Sciences. So we're very lucky today to have Oliver speaking. He then went on to do a postdoc at Oxford with Mark Wallace, and then came back to Cardiff. So a little bit outside of his formal biography. So Oliver, so going through his biography, has very, very impressive papers, good number of journals and what have you. But some things jumped to my mind. So 3D printing of Lego fluidics. That sounds very interesting. So yeah, not very often you get Lego in the titles of scientific talks. That will be brilliant. And also he has an interest in science-themed music and performed at Einstein's Garden at the Green Man Festival. So really an all-round biography and a fantastic research. And we're very lucky to have Oliver today. So on that note, I pass over to Oliver. Thank you very much. I'll just say, if you can populate the chat with questions as we go through, at the end we'll have a formal Q&A session. So thank you very much, Oliver, and I hand over to you.
Thank you very much for the introduction, Joe. Thank you. And thank you for SEB for the invitation to talk. It's great to be talking again after the award in 2016. So yeah, Joe gave a great sort of background. So I'm going to try and not just stick to science, but give you a bit of a flavour about my research journey and how I got to where I am now, really. Hopefully that will add some additional context to the journey and sort of decisions made along the way alongside my research interests. So in terms of doing that, what we've got coming up, I'll just talk a little bit about my research interests globally, how my career path has brought me to where I am, and then focus on a bit of science on the single molecule studies of membrane proteins, which is work that I began in Oxford and have carried on and sort of developed further in Cardiff, and our parallel research strands and interests, which are devices inspired by biology. So really, I suppose it's taken me a while really to really sort of distill how all my research interests fit together, because I think I'm sort of a person who follows what interests them, and there's lots of things that I found interesting, and it's sort of taken me a period into my research career to realize that really I'm quite interested in sort of concepts and tools and technologies and how these all work and fit together. So this goes from, and in my lab, that's really sort of around from single molecules and studying single molecules and how single molecules interact with each other to give rise to higher order function, particularly around the membrane, and then how we can take that learning and that knowledge that we've learned from those studies to build new sorts of devices or new tools that are inspired by biology or built out of biological components. So on the sort of tools and technology side, that includes things like 3D printing. Joe mentioned 3D printing and microfluidics, which I'll come to in a bit. So we use microfluidics to make droplets, and droplets can be used as compartments, and we can use that to study particular processes in confined or compartmentalized systems. It's also we use our droplet tool for making what we term artificial cells, models of the cell membrane and internal compartments of cells. But we also build microscopes, single molecule microscopes using lasers and optics, particular geometries that allow us to visualize single fluorescent molecules. And we do that to try and understand particularly around the membrane, how components of the membrane organize themselves, how they interact, how their structure gives rise to function. And we can understand a little bit about how features and function emerges in biological systems from their components and their interactions. And then really we're trying to take that knowledge and say, well, cells are very complicated and evolution's had a very long time to make very sophisticated structure and functions to perform certain tasks. But can we sort of take a little bit or glean a little bit from that to engineer in similar ways or ways borrowed from biology, which is kind of the realm of synthetic biology and biological engineering. So these, I mentioned, a sort of keenness on concepts really, and I think a lot of the things that excite me are kind of emergent properties. So you know, you can kind of think of as typically as humans, we engineer, we sort of like to have an understanding of what the inputs are, and we've got a vision of what the output is going to be. So how we're going to combine our materials or whatever it might be to build a structure with some certain properties that we are aware of at the outset. And biology often sort of works in, or Darwinian evolution in the reverse, in that components have properties and they're combined in different ways, and sometimes features arise. And often when our human intuition, when we're looking at those systems, is not very good at predicting at the outset what the ultimate function will be. But when we work from the function backwards, we can reverse engineer it and it makes sense. So just an example on the slide is, you know, ants and termites, just following rules of these termites building their structure of their nest. Each termite is following individual rules of the system, and this grand structure arises, which has various features like temperature regulation and air exchange, and it also has sort of shape and structural features which look very similar to sort of kind of structures that we build by design. But of course, no individual termite has a vision of what the ultimate structure is. They're just following the rules and the structure emerges from it. And we can think similarly around the membrane. And I think, you know, the membrane, I think it's very tempting when you sort of start learning about biology to think of the membrane as a barrier. It keeps the inside of the cell in and the outside out. But as you start thinking about it and studying it, you start to realize it does quite a lot more than that. And that, you know, it's made up, as you'll know, of amphiphilic lipids, so a hydrophilic head and a hydrophobic tail. And, you know, when you have multiples of these molecules, they self-assemble into bilayers, into vesicles, and ultimately, you know, these kind of structures are what make the cell membrane the lipid bilayer. And this is an example of, you know, a higher order emergent property. That individual lipid has a set of properties. When you have a collection of them, they form an organized structure that has, you know, a higher order set of properties like confinement, contain, you know, impermeable features, the ability to keep things separate or segregate things, set up gradients, things like that, which biology uses very, you know, to great effect. So we can think around our system of our artificial bilayer system, which is very simple compared to a cell. You know, it might just contain water, oil, lipid, and a couple of biological molecules that we're interested in. But these, through, you know, the lipid bilayer largely self-assembles, or we exploit self-assembly properties to make these systems. But then membranes bring around really useful properties of, as I mentioned, confinement, compartmentalization, which cells use beautifully. The membrane can help the cell control interactions in space and time by keeping things separate or mixing them, but also through reducing scales and dimensionality. There's, you know, lots of examples of reactions of molecules that in 3D don't hit a critical concentration and don't react, but they bind to a membrane which provides a surface. It orientates the molecules. You've got reduced dimensionality and assembly proceeds. So, and as well as that, you know, you just need to think of ion channels and how cells set up gradients and use that to energize processes. And you've got this sort of beautiful array of function that's arising from what we were initially just thinking of as a simple system designed just to keep the inside of the cell inside and anything external out. so in our lab, we're interested in sort of understanding and learning about those systems, and then building materials that that either are inspired by them, mimic those kind of properties, or borrow those kind of properties to perform useful tasks. And how I've sort of arrived at that position of those things being my research interest. So as Joe mentioned, I was originally a pharmacist. I qualified with a pharmacy degree, and then qualified as a practicing pharmacist. I worked in hospital pharmacy for a while, and there I kind of, was mainly working, doing all kinds of things in the hospital really, but did a lot of work on sort of radiopharmaceuticals and sterile preparation of chemotherapeutics and things like this. But ultimately, that wasn't for me. I was really interested in research, and so I went to Cardiff University to study a PhD in chemical synthesis and separations using microfluidics. So that kind of gave me a grounding in being a bit of an engineer, really, and using microfabrication tools and technologies to help with chemistry. I did a one-year postdoc in Cardiff in an industry-facing role where we were using those sort of tools and technologies of microfluidics to try and help industry solve problems in terms of chemical separations, sensing technologies, things like that. And that was before I then moved to Oxford, and I undertook a couple of different postdocs, but successively in the lab of Mark Wallace. And I was very fortunate there to be able to collaborate with Hagan Bailey and with Tony Watts on different projects. And in Mark's lab, I was sort of working on developing the droplet interface bilayer model and applying that for single molecule imaging. So when I started in Oxford, I came with my microfluidic expertise and interest in the membrane, and using those two things to try and use microfluidics to make models of the cell membrane in relatively large numbers rather than making them manually by hand. And then I moved into sort of learning about single molecule biophysics and getting involved there, really. In my final year at Oxford, I was sort of working in collaboration on a big program grant that was led by Imperial, and I had lots of opportunity to travel to Stockholm and work with Eric Tyrode there, doing some membrane measurements using some frequency generation spectroscopy. That was a lot of fun, and I think all those things equipped me well when I sort of took up my position in Cardiff. So in terms of a bit of science, I'll start with a bit of the science that I was working on in Oxford, and that has come with me to Cardiff, because some of these projects were quite long, really, in the end, and ended up taking, perhaps longer than we anticipated. And I was fortunate I was able to carry on with this work and this collaborative endeavor as I was setting up my own lab and transfer that into my lab, and we could sort of raise the ambition of the project, because it gave scope to keep working on these things beyond just a 1 year or 2 year postdoc, which was great. So single molecule imaging by turf microscopy. So I mentioned briefly about droplet interface bilayers and how we use self-assembly to assemble our bilayers. And here you can see in this figure here, this is a droplet interface bilayer on a hydrogel surface. So this is a technique that I was helping develop and further in Oxford, where essentially if you take lipid and dissolve it in oil, and then you add an aqueous droplet, the high surface tension between the water and the oil means they don't mix, and lipid self-assembles at the interface between the water and the oil, acting as a surfactant. Similarly, you can take a hydrogel layer, in this case agarose, and cover that in the same oil and lipid solution. You get exactly the same effect there, and you assemble a lipid monolayer. And by bringing the aqueous droplet and the hydrogel layer into contact, you form a bilayer where these two lipid monolayers meet. So in that technique, you're really sort of dictating the thermodynamic landscape to use the self-assembly properties of the lipid to make the ultimate structure that you want at the end, which is a model of the cell membrane. And by making it on this sort of flat agro substrate that is spin coated. It's on a very, very thin but aqueous wet layer, but it's sufficiently thin that we can interrogate it via single molecule techniques known as TIRF microscopy, total internal reflection fluorescence. So I won't go into a great deal of detail about that, but essentially we can build custom microscopes, and we've got 2 of them in Cardiff that fire a laser at a very specific, through a specific set of optics, and create a certain illumination geometry that means the laser hits the sample at an inclined angle. And because there's a refractive index between the glass and the water above it, that actually at a certain angle acts as a mirror and it reflects. And so the laser light reflects back out, but because light behaves like both a wave and a particle, it exists also above the surface it's reflecting from. So this means we can illuminate with very high intensity at the region of the bilayer where our bilayer sits. So this is perfect for being able to look at single molecule processes that are happening in the membrane. And by this specific illumination geometry, we get very, very good signal to noise. So you're not, unlike epifluorescence, you're not getting all the background fluorescence from above the sample. So you can, in fact, just see a single fluorophore. And so we applied that and continue to look at, this is an example of calcium flux imaging through a protein pore. So each of these spots that you see blinking is an individual alpha hemolysin membrane protein. And what we're looking at in the fluorescence is calcium is passing through the pore, and the other from the underside of the droplet, and within the droplet we have fluorate, which is a calcium sensitive dye. It binds the calcium that moves through the pore and lights up. And we see them blinking as the pores are getting stochastically blocked by a blocker. So the important thing to take home here is that each individual sort of spot we're looking at is a plume of calcium coming from a single ion channel, or pore in this case. And so why might you want to do that? So studying single molecules is more challenging than studying collections of molecules. You have to have techniques that can interrogate with sufficient signal to noise to be able to measure that one particular event that's occurring. And so most techniques employed in most biological sciences and in most chemical sciences involve measuring collections of molecules, either measuring their average properties or measuring their additive collective properties. But that sort of, if you have things like short-lived intermediates, or you have variations in behavior or different pathways, your measurement is just averaging all of these out, and you can't reveal exactly what's going on. So one example might be, an analogy being, cars, for example. If you take this sort of time-lapse view of a city, you get to see where the roads are, where the light is coming from the vehicles. You can make some inferences like here's brighter than over here. So, but what we can't tell from that is that more cars, or is that the same number of cars, but they're stopped for longer and therefore you measure light for longer. So these kind of problems become difficult to unpick if you can only measure the ensemble average. But if you're luckily cars are big, so you can just look and count the cars, or you can track the cars, but you can unpick that question quite easily because you can look at individual vehicles. So we're trying to think about in the same way around the membrane. If we can track the structure and function of individual membrane proteins, we can start to understand their function at a level that is otherwise inaccessible. And perhaps a very pertinent example of single molecule sensing is from Oxford Nanopore. And so this is a spin out from Hagen Bailey, and I was very fortunate to be able to work and collaborate with Hagen Bailey, and still get a lot of inspiration from my time working with him. And every time I speak to him, he's a very inspirational person. If you're not familiar, the nanopore sequencing that Oxford Nanopore sort of specialize in, they're pulling a strand of DNA through a protein pore and measuring the electrical current of ions passing through this pore at the same time. And so as the ions pass through the pore, which is measured via electrophysiology, and then as different bases of the DNA strand occlude the pore to different extents due to the different shape of their chemistry, you measure a different current for each different base combination, and then that can be processed and turned into your DNA sequence. So this is a wonderful technology and a really impressive example of how single molecule analysis can have real important implications. And so in the work that I did in collaboration with Mark Wallace and Hagen Bailey and Shou Huang, who's now in China, we were trying to see if this technique, if we could do the same type of measurement optically. And so we've got this droplet interface bilayer system and imaging set up, and we have our alpha hemolysin pore and our DNA, which we've capped with a streptavidin. And under an applied potential, we can pull the DNA into the pore, as you see here. And if we reverse the potential, because DNA is negatively charged, we eject the DNA from the pore. And so we were hoping we could, with these different levels of blocking, firstly from open and not blocked and blocked, but then from different bases of the DNA, could we measure that from the optical signal that was detected via turf microscopy that was coming through the pore. And so that in practice looked like this. So here's our individual pore, and we can turn it off or turn it on, open it, trap a DNA and read out what its base is. And we can repeat that and do it again, trap a different strand and measure what the identity of its base in the trapping region. And so with that, we were able to demonstrate that optically we could distinguish between A, T, C, and G bases residing in the pore. And you might say, well, why do you want to do this given you can already do it electrically? And I guess the answer there is that optically, as you can see from imaging, you're imaging multiple pores at the same time. So it's a way of scaling the measurement that you could electrophysiologically, you can only measure, if you have two pores in your bilayer, then you just measure the sum additive total of the current through both of those pores. So this gives you a way to look at multiple pores simultaneously and potentially provides a route to scaling. But I think also of particular interest to me was that you can get the kind of resolution that you can discriminate those small differences, and that's something that then can be applied in a physiological situation to unpick differences in function of ion channels, for example. So that was some of my postdoc work in Oxford, which, by the time it was finished, I was already in Cardiff. But I was fortunate enough to be able to get an independent fellowship at Cardiff, and this was a Chancellor's Fellowship scheme, and that allowed me to set up my own lab. So I moved from Oxford back to Cardiff as it happened. I didn't necessarily always envisage or plan to come back to Cardiff, but it was obviously on my radar as a place to go, and an opportunity arose, and I was able to establish my own lab, which was a really great opportunity there. So when I started, I was faced with essentially an empty room with no equipment and anything like this. So something that was vital there was I was able to maintain the collaborative links that I'd already established in Oxford. Oxford was a really sort of stimulating place for this, and getting lots of interesting exposure to lots of interesting science and interesting people. And I'd been working on the program grant that was led by Imperial, and I was able to sort of continue to be affiliated to that in a non-funded capacity, which allowed me to sort of have a big network as I started. So I didn't have much in the way of equipment or facilities. I had to build all that from scratch, but I felt like I had momentum with ongoing work and collaborators that I could work with, which was really helpful. And so one of the key sort of pieces of work that started in Oxford and then continued in Cardiff, and was quite a long piece of work by the end, and this was with Mark Wallace and Tony Watts, and Alan Goddard, who's now at Aston, Patricia Dickman, who moved to Germany and is now in industry. So I was on the single molecule experimentation, analysis, and modeling side of this. Alan and Patricia on the protein expression purification, but we were really interested in GPCR dimerization. So at the time, there was a lot of I guess, dispute in the field about, there's 800 or more human GPCRs, and then they're studied to varying degrees, and there's often a lot of assumption that one thing that's learned about one GPCR is that transferable to the whole class of GPCRs. And something particularly important here is dimerization. So GPCRs, there are some that form permanent dimers. There are others that there's evidence that they form dimers, and also evidence that they go around as monomers. And there's quite a lot of conflicting evidence, and a lot of that, I'll get to why that is. But there were sort of two schools of thought about, are these GPCRs, and in this case it's the neurotensin receptor NTS1, is it functional as a dimer or is it functional as a monomer? And so we used, the idea was to use this setup. So I've now progressed, got a, this microscopy setup built in Cardiff, which is for the single molecule experiments previously discussed were showing ion flux through a membrane spanning pore. Here we're looking at individually labeled receptors and how they move, and we're using FRET, which enables us to see if they come together. So if two species labeled with donor and receptor come together, we'll get a emission from the acceptor fluorophore, which otherwise we won't see. And we can capture that on camera. So here we're looking at the same portion of the membrane. Each spot you see is an individual protein diffusing around. On the right-hand side is monomeric protein, which is labeled with Cy3. And on the right-hand side, we only ever see, this is the same portion of the bilayer, but we're imaging at a different wavelength. So we only ever see a spot here if we have a dimer form and FRET occurs. So you can see these spots occurring, they arise, they disappear, but notably there's a lot less, they're a lot fewer than the monomeric side. We can then track these proteins and extract diffusion coefficients from them. We can see that what we're assigning as a dimeric species diffuses more slowly than the monomeric species, and that's in line with an increase in doubling of the hydrodynamic radius, which would make sense for a dimer. So that's sort of theoretically consistent. But importantly from this, we could analyze these single molecule images with quantitative image analysis techniques. I haven't got time to go into the detail there, but essentially you can pull numbers out about how these things diffuse, how they interact, the frequency at which the new dimers are observed to form, how long they're observed to last. And from that sort of statistical information, I was able to build a Monte Carlo simulation model, so we could extract with precision and accuracy at the single molecule level how these dimerization interactions arise and occur. And I think an important thing is that we were able to characterize that they're short-lived. So the molecules come together, but then they only stay together for hundreds of milliseconds to seconds before they come apart again. And so you can see very quickly, and from running the simulation, you can see that then it occurs that, well, the amount of dimer that you have is really strongly dependent on the surface density of the protein, because if that doesn't stay together as a dimer for very long and it comes apart, you're really reliant on how frequently they collide. So if your surface is very dense with protein, they'll collide very frequently and you'll have a lot of dimers. And if they're sparse, they collide infrequently, the dimers don't last very long, so they fall apart and most of your population exists as a monomer. And so from being able to actively measure this at the single molecule level and then perform simulations, we were able to show that perhaps these two schools of thought around dimerization about permanent, it exists as a dimer, it exists as a monomer, are both right, because essentially it depends on the experiment. And I guess this is an important thing to think about unforeseen artifacts of your choice of experiment, in that if you're overexpressing the protein in its cell, for example, the cell is then jam-packed with protein, collisions are more frequent, you get more dimer. If you express the protein, purify it, and put it in proteoliposomes, and you're measuring on the ensemble level, so you need a high signal, so you try and put a lot of protein in, again, you're measuring mainly dimer. If you're looking in other systems, natively expressed systems, you might find that it's mainly monomer because the density on the cell membrane is a lot sparser. So both answers can be right, and that is explained by what was found in the single molecule studies. And interestingly, the cell exists in this region here, which is a very steep portion of the curve, which, it's tempting to speculate why that might be. So perhaps that means the cell can, corral the protein or change the density, and therefore change quite readily the amount of monomer or the amount of dimer. And if, for example, electrostatic changes or other changes affect how long the dimer stays together, that's another route, if you're in that sensitive region here, to govern whether you have mainly dimeric or mainly monomeric species. So that raises then sort of obvious questions about, well, what are the functional implications? We did some work on looking at, what's the conformation of the dimer, and from looking at the intensity versus some standards, you can extract an average distance. But interestingly, that average distance doesn't correspond to any particular single interface. And what we see is this kind of individual dimers fluctuating in their FRET efficiency. So this is essentially meaning that the closer the two fluorophores are together, the more efficient the energy transfer, the higher the signal in the acceptor channel. The further apart they are, the lower the signal in the acceptor channel. And this scales to the power of 6, so it's a very sensitive distance measurement technique. But we see these forays into high FRET states, then to low FRET states, high FRET, low FRET. So that's strongly suggestive that the interface of the dimer is dynamic and it can move around, which is really interesting. So, that sort of fits in with some other observations that we made, such as, the collisions were seen to be very effective, a high percentage, about 25% of collisions leading to dimers. If you only had one productive interface, you would expect that to be a lot lower because the two interfaces would have to hit at the right orientation. So, some more interesting things that we were able to unpick by studying at the single molecule level. From that comes what are the functional implications, and we sort of corroborated this with ensemble techniques and molecular modeling. And the functional implications aren't that clear, but we think that in the conformations that are most likely from the FRET distances and the modeling, that ligand binding and G protein binding is precluded in certain conformations and encouraged in others. So that gives sort of possible leads for future study. But we, that was our take-home message and what we were able to reveal from these single molecule measurements. And I mentioned about this being work that started in Oxford and then continued in Cardiff. And something I want to sort of say is that, science can take a long time. So this originally started as a 2-year postdoc, and it ultimately took 7 years. So there was a lot of, it went a lot further than was originally anticipated. And I'm, very grateful to Mark Wallace and Tony Watts that they really let myself and Alan, who was at Lincoln at the time, really drive this, because we were working on this as postdocs, and then we moved to our respective institutions, and we were really able to drive the rest of this study for the other 5 years of it, and get to the point where the knowledge was. So that was really useful and very helpful for me. But it also sort of opened my eyes, I guess, to the sense that science can take a very long time. And when you're starting, when you're starting out, or when you're starting your lab, or if you're in a postdoc position, you can need some quick wins. And so I sort of tried to develop a strategy of doing some fast science alongside my slow science, so that I could get papers out while working on these bigger projects, for example. And that's where some of the 3D printed Lego microfluidics come in. So some of the other things that we've done in my lab in Cardiff and are doing, 3D printed microfluidics. So here we were taking just, off the shelf fused filament 3D printers and using them as an alternative to traditional microfabrication techniques. So 3D printing is incredibly quick. So you can make something, you can redesign it, you can test it, you can iterate, you can send designs to other people. It's got a lot of advantages over more traditional fabrication processes for rapid prototyping and development. So we made these modular microfluidic systems that we could combine in different ways because they were Lego-like to make different modular structures. And this was just a sort of point in that there's, at the time, we found it quite hard to get this published. We sort of went around quite a lot of journals and got reviews back, and the reviews were often, you know, why would you do this? Because you'll get a better result if you use a more traditional, more accurate approach. And I think the message here really was that, yes, there are higher fidelity ways of doing this, but this has its own advantages in that it's very quick, it's very easy, it's very cheap, and 3D printing is going to grow and grow and grow. And I think that's been borne out in that this is, you know, one of my most highly cited papers, but at the time we really struggled to get it published. And so I think that just shows that you know, the where your work ends up in terms of papers is, it can seem very important at times, but really sometimes it's you know, the prevailing wisdom at the time isn't always correct, and what wasn't necessarily deemed to be interesting, you know, you can think it's very interesting and you can back it, and keep backing it, because, you know, ultimately, as long as it's out there, people will find it and cite it. And so this got quite a lot of interest, and we had various opportunities arising from this with Ultimaker. And now 3D printers have advanced in the intervening years, that 3D printing now is our mainstay of producing microfabricated devices and microfluidics. So that's really advanced. Then I mentioned earlier on about how, you know, I've been talking about the science of single molecule studies of membranes, but also how these membrane approaches and the development of those membrane platforms as tools to understand the membrane and membrane biology, we're also repurposing those as functional materials in themselves. And we sort of term this artificial cells, and that's in the sort of synthetic biology world. So we're doing and have done a lot of work on that. We work on biohybrid materials. So in collaboration with David Jones here in Cardiff, we've been decorating carbon nanotubes with proteins, and interestingly, you can measure properties of the protein by electrical conductance of the nanotube, but also you can dock fluorescent proteins onto these nanotubes and measure the fluorescence, and the fluorescence properties change with the electrical state of the nanotube. So we've been developing this towards an optical readout sensor that can, you know, look at protein. You can use GFP, for example, coupled to these systems as a reporter for other proteins which aren't inherently fluorescent, and study them at the single molecule level. COVID was a big disruption for everyone, and worse. But in Cardiff, I was amongst a sort of team of people that we very early on in the pandemic, we set up our own PCR testing pipeline, and I was involved in that. And further opportunities came from that in terms of we developed a CRISPR-based assay for looking at variant-specific detection. So we applied that to our pipeline of PCR testing. We applied it also to wastewater monitoring, and you can see the different variants. So if you're, you know, this is obviously applicable to any pathogen, because, you know, they all have their genetic code. But the ability to go from you know, PCR, where you're identifying the pathogen, but being able to, without sequencing, say what variant it is, down to single point mutations with something. So as long as you know the mutations, as long as it's been sequenced somewhere by someone, you can then pick that up in a molecular test, which is much more portable and much more immediate than waiting for sequencing. So these are some of the other things that we've been worked on. I'm conscious of time, but I'm just going to sort of talk about the artificial cell work that we've been doing. And I've been sort of fortunate to be involved. Cardiff Trento were leading, and Cardiff were were equivalent standing, but we weren't leading due to difficulties around Brexit. In this EU project around artificial cells, and sort of the concept being these artificial cell materials that I've discussed based around membranes, based around compartments. We've got a feeling that, that biology uses those things very, very well and very, very nicely. And there's a lot of advantages over, you can do chemistry in compartments that you can't do in a single pot. You can start to control how different interactions are isolated from each other, how they communicate with each other. You can set up sequences. You can also set up parallel processes, potentially only activate the one you need, for example. And because these compartments can be networked and they can talk to each other through membrane proteins, we've got quite a sort of adaptable platform technology that can be used in different ways as we develop more and more sort of molecular level control to implement different processes within this chassis. So we're seeking to sort of apply that for applied situations such as drug delivery or screening, high throughput screening, application outside of the lab. So one of the main challenges there really is, it's all very well me talking about these droplets of water and oil surrounded by a lipid monolayer, but that's inherently, something that you make in the lab. Is not very stable and it's in an oil environment. So we developed some microfluidic systems. I don't really feel I've got the time to go into lots of detail here, but essentially using droplet microfluidics to make these droplet interface bilayer droplet network systems and then wrap them up in a hydrogel shell. And by wrapping them in a hydrogel shell, they then become aqueous compatible. It provides some structural rigidity. You can move them, you can manipulate them, you can pipette them, you can pick them up with tweezers. We can insert membrane proteins into them. So here we're inserting membrane spanning channels and interrogating via electrophysiology. And you'll notice here, we've got in this diagram, we've got 3 cores. We can insert those membrane proteins either in the membrane that exists between those cores and the outside world, or between those cores inside. So that gives means of communicating via those membrane spanning channels with the outside world, or between those processes in those separate droplets within inside the material. So this is just showing we can regulate chemical reactions inside those. We're just looking at some bioluminescence here, but that can be energized within those droplet compartments. So we're starting to energize our artificial cells. We can look at use them for high throughput screening. So here we're using microfluidics and the fact that allows us to make lots and lots and lots of these artificial cells to be able to screen different conditions of 2 synergistic pore forming species and try and understand how they work together when they give rise to elevated function in what combinations, and when the activity isn't enhanced. I don't have time to go into the detail of this, but this is just saying that our multi-material 3D printing has really expanded. So we're now 3D printing multiple materials in the same device to make these complex artificial cell emulsions that we can effectively print out of a single head. And by using what 3D printing enables, which is 3D geometries, we can really control the spatial organization of these structures, how many compartments we have, how they're organized and ordered, where the membranes are. And with advanced microfluidics that Jin Lee, who's been an excellent postdoc on our European project and now has an independent position in Cardiff in the School of Engineering, he's really driven programmability of these systems. So we can program the microfluidics, the geometries that are made, the flow profile of the microfluidic device, to program the sequence of droplets and how they assemble in the artificial cell. Because as I was mentioning about the contents and their connectivity gives rise to the function. So being able to control exactly what's in them and exactly what order they're connected will allow us to make, it will dictate the function of these artificial cells. Because if you want a sequence specific reaction, you need those compartments to be connected in the appropriate order. I'm about to wrap up, but I'm just going to show you how we've done that in terms of using acoustic levitation to levitate these artificial cells. So we can produce them microfluidically, trap them in an acoustic standing wave so that they levitate, and then we can study them and image them and manipulate them in this environment. And we've used that to create them and post-process them, but we can also activate individual membrane proteins in this system. So here, this is the ion channel MSCL, which we can modulate the acoustic field and a superimposed magnetic field to trap and frustrate the droplet, to introduce membrane tension, and then activate the MSCL ion channel. So MSCL is activated, is a release channel in bacteria. It opens in response to osmotic pressure or stress or mechanical stimulation. And we can use that to selectively open our channels of communication between different droplets in our network to trigger subsequent reactions. And because it's being triggered by the acoustic effect, we can turn that on and off. So we can effectively have a remote way of dialing in function into our artificial cells that we can say, okay, activate this channel and it will open the channel, but we can also close it again, which, unlike stimulation with a chemical species, is that once you've added the chemical, you've added it. Okay, so this is a way that we're trying to get our artificial cells to interface with the outside world. And on that note, I think I'll just leave you with where we're going with this, which is we're now taking those artificial cells and interfacing them with live cells. And we've done some more work on Lego, which we've recently printed. And this started as a series of undergraduate projects, actually. And alongside Ahmed Mukhtar's PhD, he was really able to sort of help guide and drive those undergraduate projects. But we decided that, you know, we could build a bioprinter very cheaply from the robotics of Lego. So Lego make these programmable units. And again, this sort of feeds into the, you know, you can do very expensive science. We build these microscopes for imaging single molecules, which cost multiple hundreds of thousands of pounds. But you can also be resourceful and use things that are cheap to do quite sophisticated things. So we built this programmable bioprinter that we were able to print droplets that encapsulated cells, deposit them in defined places to build up three-dimensional tissue models. And we've just learned that we've won. So I'm leading a consortium, an EU consortium, in the next round of the EIC Pathfinder. And we just learned that we won this project called BioHost, which is working with colleagues in Trento and Zurich to sort of further advance our work on artificial cells and combining them with living cells to make living functional materials. And so that's the trajectory that we're going. That was rather whistle stop, but that brings me back to this summary of how really that this sort of fits in terms of tools and technologies, using those tools and technologies to gain fundamental understanding around the membrane, but also taking that fundamental understanding and building tools inspired by biology that we hope will see applications in the, you know, that a lot often these things aren't near-term applications. These are sort of quite a different way of thinking about engineering materials. So applications in healthcare are going to, and tissue engineering and things are going to be a while off, but they're in the nearer term, they're making laboratory tools that enable us to increase our sophistication of study of, you know, of being able to better replicate tissue models in the lab, or being able to have better models for understanding biological processes. So we see sort of near-term application in research, and then longer-term application in sort of wider world, real-world applications for these artificial cell, artificial tissue type materials. And so that's me. That's done. That's just like to thank so my team in Cardiff, collaborators in Cardiff, collaborators on our European grant, collaborators we work with on the COVID testing, collaborators from Oxford University, and particularly Mark Wallace, Hagen Bailey, and Tony Watts, and our funders. So thank you, and also to SEB.
Fantastic. Wow, that was a really, really fantastic talk. Thank you very much for that, Oliver. So if anyone has any questions, if you could populate them in the chat box, and then we'll go through them. But I will take chair's privilege and ask a few questions early first. So I was really, really interested in your threat imaging between your two different. So when you were showing the threat efficiencies between the dimers and the monomers, and it got me thinking, have you done any work with crowding with relation to other proteins? So other things which are in the membrane, and whether that would reduce or increase the dimerization efficiency? Because at first I thought it would reduce because they wouldn't be moving around as much, more crowded environment. But then I wondered whether, oh, well, actually, if they find each other, so if you think about in a normal cell, they're around a lot, but then as soon as they find each other, they stick to each other, and efficiency, there might be a higher proportion of dimers, if that makes sense.
Yeah, that's a really excellent question. So we haven't is the answer there. And so this is in sort of the most minimal functional system. So we've got one single lipid and then just NTS1 protein labeled as two populations. And so I think there's merits both ways. Okay, so from having, so I suppose where we are at the moment is we know that this is a fundamental property that emerges from this small palette of components, and it can produce the potential for quite sophisticated function, because if you only need different types of biases to either change the amount of dimer that you have, change the preferred interface. And if I mentioned very briefly, but I didn't have time to go into detail, but when ligand binds, you get a conformational change, and that has some allostery then with the what dimer conformations are likely or less likely. So very quickly you start to get a lot of combinatorial behavior becomes possible. And so I think your question, Joe, is an excellent one, because it gives exactly what to do next, which is make the environment more complex, or add these things in one at a time, do it with ligand, do it with G protein, and see how this biases the system. And perhaps one of the strengths about doing it in a very minimal system rather than a cell is that you can add these components one at a time, or you can make an approximation of these things one at a time and look at the effect. And I think often, so for when we were sort of, I think often purely in vitro systems sometimes get a bit of a bad name because it isn't in the cell. But I think as long as you're thinking about what does this tell me about what could be happening in the cell, or what lens does this give me to view the cell through in terms of what is fundamentally possible from the properties of this system.
Okay, so I published. So I'm a plant cell biologist. I published some work in 2019 using single molecule imaging in plant cells, and we were looking at membrane protein, and we basically found out the cell wall was super, so the extracellular matrix was super important for the diffusion dynamics. So as soon as we found some weird things with actin and microtubules. They seem to affect different proteins that we looked at differently, but the cell wall just completely, which makes sense because biophysically it's pressing down on these things. So so that's where I was going with that. And so I'm fully on board with 3D printing. I'm a lover of 3D printing, but just maybe a quick, and it's a lot quicker, it's a lot easier, what have you, but what can you maybe comment on the robustness of the parts you make compared to a commercial alternative? Like, should you expect to make 3, 4 for every commercial one that you buy? I still think you should be making the 3D printed parts, but just wondered, from how robust they are.
Yeah, that's a really good question. So I'll answer that specifically with regard to microfluidics. We use the 3D printer for all kinds of stuff in the lab, building like optical holders and all kinds of things. So I would like really strongly encourage anyone to get one if you're working in practical lab science, because you can find so many sort of practical shortcuts of things that you just help you do your work. In answer to your question, Joe, there is some. I think this is something that we've got a lot better with time, and I think part of that is the printers have improved massively, and the other part is that our skill has improved. And but I think at first we were probably finding that the most common problem was that the materials we were printing, it might lift off the build plate and therefore it starts to warp. So I think if visually you see that the printing process has happened as you expect, it's still on the build plate and it hasn't warped, then it's usually good and reproducible. One of the key challenges for making fluidics is optimizing the extrusion rate and the density of fill, so that if you under extrude, you essentially have gaps, and if you over extrude, then you lose fidelity of your channel geometry. So it took us a while, and we reported in the paper all of the settings and things that we used, but really that's the thing to optimize, is that you get the packing of the material as it's printed right, and then you can make a very good device. I'd say the biggest difference is not so much because I suppose we do probably, I would say maybe, I don't know, 1 in 4 doesn't work as we would like and you throw it away, but the easy thing is you can just make another one. And so it's not quite got the same sort of overhead. And I think often, if you do your experiment and then think, oh, I need to change that slightly, or I've got another idea that I want to test, you can just change your design and make it again. Whereas from a commercial supplier, depending on how they make it, that's a very expensive and long process. So, and I think we've recently in Cardiff got with Jin Lee, who I mentioned, we've got a new 3D printer that can print down to 5 micron resolution, but it can still print big structures. So formerly 2 photon printing was usually limited to very small areas, and this has got the ability to do big areas but also small features. So now that's closing the gap in terms of the channel geometries. With the fused filament, we were essentially about the smallest geometry channel you can make is about 100 microns if you've really optimized all the settings. But you can do some tricks. We can make droplets down to about 20 microns using fused filament printing. So it's kind of it's getting better and better all the time. And I think the speed advantages really outweigh those other ones.
Wonderful, fantastic. And so I think time-wise, just one last question. Sure. Science was phenomenal. Thank you very much. What I really liked is the way you spoke and gave some advice going through the talk. From you go to conferences, you see these amazing talks, brilliant science, and you think, oh my word, how am I ever going to compete with that? I really like your idea of fast science alongside slow science. Very good, makes sense. Science takes time. Thank you for pointing that out. But I wondered if you had any advice for early career scientists trying to get postdocs or permanent positions. If there are any, if you could give 1 or 2 top tips for people that are making that difficult transition, what would you say is important?
That's really good question. One of the first things I'd say is that you can't follow everyone's advice. So lots of people will have advice, and you'll see lots of people who've done good things and don't feel pressured that you have to do all of those things, because that's ultimately impossible. And I think that's a trap that people kind of fall into, thinking that, oh, so-and-so said I should do this, and someone else said I should do this, and someone else said I should do this. And there's only so much that you can do. So recognize that. So my sort of tip would be, apply for things. Don't be afraid to take risks in applying for things. I see in Joe, I bet you probably think similarly, moving from a position of being the person who's applying for things to being the person who's advertising jobs, all of a sudden you're thinking, I hope some good people apply. And when you're applying for jobs, you're thinking, oh, I bet loads of good people are applying, I haven't got a chance. And it's funny how exactly the same scenario, your mindset totally shifts when you're on the other side of it. So that, and I would also say, if your institution has any, if you're a PhD student or a postdoc, if your institution has any sources of funding that you can apply for, I think Cardiff University has been particularly good at this in that it's had Wellcome Trust sort of strategic awards, which it's allowed postdocs to be principal investigators on those internal bids. And that really does give you a track record of funding, the opportunity to start developing your ideas, understanding how the grant writing process works. And I think, you know, and then my final sort of tip, given time, would be that, you know, there's a lot, when you're in that postdoc situation, I know a lot of people feel things feel very dictated by chance and timing. And you know, your funding's coming to an end and you need something else. And I don't really have any particular solutions to that, but that's a problem. And I think, you know, increasingly PIs recognize these challenges. And you know, so if you've got opportunities to build relationships with people or labs that you'd like to go to, and the timing might not be right now, but work with people, because it's always really good to have people who want to come to your lab as well. And you know, I've just somebody wants to come to my lab. We've just submitted a Marie Curie fellowship today, the deadline is. So, you know, so if there's someone whose science you like, engage, talk to them. You know, if it's a fellowship opportunity, you're doing a lot of the work there, and people are usually glad to say, yes, I'll support that, because, you know, it's if you get on and you've got your ideas align, then that's really good. So just put yourself out there, be prepared to take risks. Don't get too, only allow yourself to get as down about things as you allow yourself to get happy about things.
Fantastic, brilliant. Well, in the sakes of time, I think we'll call it a closer. I am virtually clapping and saying, yeah, really, Oliver, thank you very much. I really enjoyed that. It's a really good talk, and I really like the narrative you built and interweave it with your career path and what have you. I think it's really useful for everyone that's been on the talk. So, you know, thank you very much for your time, and we really appreciate it. And thank you everyone for attending too. So yeah, and there'll be more of these talks and more things going on. So please stay tuned and look out for more SCB events. Thank you very much, everyone. Bye.
Thank you.